Hi, this is Chris with MDPI Encyclopedia, and today we're delighted to welcome Professor Dr. Daniel Rittschof, an editorial board member at the Journal of Marine Science and Engineering, who's going to talk to us about his research on marine microbes. Welcome, Professor.
Nice to be here. Thank you for having me.
It's great to have you. So, to begin, our audience would like to know more about your academic journey. Could you share a brief overview of your research background and what drew you to studying marine microbiomes and molecular biology?
Okay, so marine microbiomes and molecular biology are a small part of my portfolio. Um, I'm trained in terrestrial ecology from the University of Michigan. While I was doing that for fun, I worked in a cell biology laboratory at the University of Michigan and took a job in biochemistry at the University of California, Riverside, because I wanted to be a chemical ecologist. And when I thought about chemical ecology, that meant water. And water for chemistry means biochemistry, not natural products chemistry. And I was, and still am not a scholar. And one of the benefits of not being a scholar is you don't sit in silos because you follow questions and you go wherever they want, wherever they take you. So questions that I asked in 1978 are being answered by omics now. Um, and omics wasn't developed then, so it took a really long time. And to show you my naivete, uh, it never occurred to me that biochemists worship clear solutions. Routinely, there's one nice answer. And that I found that very elegant. But ecologists worship variation and predation, competition, and exclusion. And they're fundamentally a totally different culture. So they really don't respect each other. By and large, the majority of ecologists don't like biochemists and vice versa. Um, they just don't think they're real scientists. So when I tried to become a chemical ecologist with biochemical training and ecology training, people told me I didn't know what I wanted to do. And so I asked some questions when I was a biochemist, masquerading as a physiologist at UCLA, um, about chemical signaling in marine systems. And I started those questions with a system where hermit crabs, who were size 10 animals in size 8 shells, can find a new shell at a predation event. So when a big snail eats a little snail, the hermit crabs, from the signals that are being released from that meal, know what snail is dying. They know what species of snail is dying. So it's a very, very specific cue. And I made a couple of intellectual mistakes that were a gold mine. I picked the wrong Nobel Prize winner as a joke. Didn't realize it was the wrong one. And found out about an enzyme called trypsin, the oldest enzyme known and in every organism ever tested. And it turns out that the signals were being made by a chainsaw radula of the big snail lubricated with trypsin. And the pieces that came off end in two basic amino acids. Those same peptides make white blood cells creep in your body. They're generated during the complement cascade, which is activation of the innate immune response and blood clotting. And they're signal molecules in marine systems between animals as well as within animals. And virtually every phylum of marine animals uses those external signals to do things, to resource locate, to know where to settle, to find prey, um, to induce metamorphosis, all sorts of different kinds of functions. A symbiotic anemone uses those molecules. Arginine vasopressin is one that's in humans. I can put that in a microscope slide and make a symbiotic anemone do somersaults on the slide because it's going to reattach to a hermit crab shell. So it sticks its tentacles on the slide, grabs on, lets its pedal disc go, and then does a somersault onto the shell. And the natural molecules actually come from the proteins that are in the shell. So I started that question in 1978. I've been very, very lucky because it's a huge chasm between biochemistry and ecology. And biochemists don't want to be in that chasm, and ecologists don't want to be in that chasm. So I've had it to myself for 40 plus years. It's lonely at one level. At other levels, it's much more fun because there's cooperation when I work with people, not competition.
Mm.
I find ultra competition not healthy for
Mm.
science in general. So I masqueraded as a physiologist when I couldn't get a job as a chemical ecologist. Then I got hired at the University of Delaware as a chemist to help identify molecules that oyster drills use to find oysters. Turns out oyster drills, when they're babies, crawl away babies, they're a millimeter high, can creep for 2 meters before they starve to death. And they only locate one kind of prey, and it's barnacles. And it's using the odor of the barnacle to find it. Barnacles are stuck on surfaces, they can't go anywhere. And the snails find the barnacles by their body odors. Well, just by chance, not at all by chance, by evolution, the barnacle body odor molecules that snails use are the same molecules that barnacles use to tell other barnacles to set near them because they're sexual animals, but they're standing on their head. And in order to reproduce, they have to have other barnacles nearby to have sex with. So what's happened is the predatory snail has tapped into the settlement pheromone for barnacles. And I actually know when I started out, I wanted to know things about these molecules. I wanted to know what molecules made animals do things. And every system I looked at that was a life or death system, like the one I just described, hermit crabs need shells. If they don't have the right shell, they can't reproduce, they can't have sex. And if they have the wrong size shell, they get eaten or they die from environmental issues. So everything I was looking at was life or death. And it never occurred to me that that's what the innate immune response is all about, is life or death. Bacteria are trying to recycle you. And if they're too good at it, we call them pathogens. So they only can be good enough to recycle you when it's your time to go, not when they want you to go. So as I've gone along, I've learned that recycling materials is really, really critical to communication in marine environments because the pieces get away. In order for bacteria to eat something like glue, it has to secrete enzymes. And when it does that, some of those pieces get away. and they say barnacle, or they say shell, or they say something else really precisely at nanomolar to femtomolar amounts. So they're pheromones and signals that are generated by enzymes on the outside. So my interest in bacteria is because in a lot of systems, they're the ones that are producing the enzymes because they're the recyclers. In other systems that I'm working at, um, oh, say you're a stone crab. Stone crabs are nice commercial crabs. They're big. They got really strong arms. Any stone crab I know I can hold by the claw, and it can do that and lift 4 people off the ground. So they're very strong. They're tough looking. And they're very scary to things like hermit crabs because their body odor says stone crab, and that's like lion. And the stone crabs use enzymes to clean their gills. Lots of marine animals use enzymes to clean where they can't scratch. So, crabs that brood eggs, like lobster and blue crabs, things like that, secrete enzymes on those eggs to clear the microbes off the eggs.
Mm.
If you take the eggs off the crab, they foul and die. So there's lots and lots of enzymes being used in this kind of warfare. When barnacles settle, one of the first things they do is kill every bacteria under them. And as they grow, they actually clean the surface with a detergent and then kill everything with reactive oxygen species.
Mm.
So their glue surface is free of living organisms. That's what
Mm.
makes a good glue, is you don't want it being broken down.
Mm.
And their glue curing is the same mechanisms as blood clotting. So their glue curing is they basically wound themselves, bleed, and then rework the scab into shell and tissue.
Mm.
So that's the kind of stuff I do. I work on glue curing, I work on communication,
Mm.
and I work now on what I call functional microbiomes,
Mm.
which are all of the enzymes and products that are related to bacteria and their hosts.
JMSE is, of course, a leading journal in marine science and engineering. So, what motivated you to join its editorial board, and how do you see it contributing to advancements in understanding marine microbiome?
Okay, so you can look at me and tell that I didn't get in this business to be rich and famous. And part of there are two obligations that people that are lucky enough to be scientists really should meet. One of them is reporting what they've done, and the other is supporting the research community. So I joined the editorial staff to support the research community. Uh, and it's probably my reviewing and editing are actually the only reading I actually do because I write 6 to 8 hours a day. And the rest of the time I'm reviewing for journals and doing editorial stuff for journals. Um, and after, I'm still not a good writer. I've only been writing since 1972, 68, around in there, but I'm getting better at it. Uh, and so I do a lot of writing. I do a lot of editing. Um, I'm pretty good at converting a variety of dialects into standard English. Um, I feel really lucky that English is my first language because I have a leg up on the rest of the world that doesn't have English as a first language because that's the language of science. So I join editorial boards and I do reviewing to meet my obligations to the community.
Mm, absolutely. And picking up on something you started to mention just before, functional microbiomes play such a crucial role in marine ecosystems. So, what key unresolved questions about their organization and interactions drive you and your current research?
Okay, so if you think about molecular biology right now and metagenomics, to me, the use of metagenomics to describe bacteria is like taking a class picture. You know who's there. You don't know who interacts with who. You don't know who's the mover and shaker. You don't know who's the socialite. You don't know anything about that community. All you know is who's there, and you don't know what they're doing. And so one of the things right now that I'm pursuing with respect to that is the families of enzymes that are associated with uh, those microbiomes. So I don't know who's putting out the enzymes, but what I can do is give a microbiome a bath in pure protein for 20 minutes. And if I do that, I get about 12,000 peptides by shotgun proteomics that were generated from the pure protein. And then by looking at the ends of those peptides and their absolute sequences, because they're pure proteins, I can know which endoprotease cut them. And it turns out when I give things like ovigerous barnacles or ovigerous pea crabs, animals with eggs, a bath like that, I get their microbiome plus I get their secretions. And there are 14 families of proteases that are generating the peptides. And I know when eggs hatch that all of those enzymes don't change in their relative proportions. So to cut the glues, the embryos and the mother have to be putting on a second substance because the enzymes are constitutive. They're different than background seawater, but they don't change on hatching, on egg hatching. So, and I'm sure the same is true for the different enzymes that cut sugars. And for the different enzymes that cut DNA, everything is being recycled. And the pieces that are getting away are what marine organisms use to organize their communities. Oysters have specific odors, and they tell other organisms they're there. And other organisms locate the oysters because that's where they live or that's who they eat. And that goes from the microbial level to fish. Humans aren't very good at detecting those odors
Mm,
because they use them for signals on the inside. The molecules I work with are represented by 31 families of hormones in your body. And one of the things I do is take advantage of the medical literature because people care about people. And the medical literature is about 25 years ahead of the marine literature. And in my area, there's billions of dollars spent on people doing endocrinology and immunology in humans. There are probably $100,000, $150,000 spent on a year on people working with the same systems on the outside of organisms.
recent advances in molecular biology, for example, metagenomics, single-cell sequencing, have transformed marine microbiome studies. So, how have these tools reshaped our understanding of microbial functions in marine communities?
They're not shaping them yet,
Mm.
um, because very, very few people um, have the resources to actually do that kind of work. I think that probably the best work is done by JMSE in Singapore. That's a big institute used to be run by a guy named Stefan Kellerberg, um, who was Swedish and then he was Australian and then he moved to Singapore. So there's a tremendous amount of work that's done just on microbiomes and major advances in bacterial microbiomes. The interaction of those microbiomes with metazoans is basically unstudied. And the people that are studying it are finding out that the innate immune response is important at a variety of levels. Things like um, marine animals using something like an O antigen to decide where to metamorphose, or people using a peptide that's generated by or organisms using peptides generated by the complement cascade being forced to settle by peptides from that. So there are edges of it, but the area is almost wide open because people are in their silos. Microbiome people study microbes, macrobiome people study macrobes. Very few people cross over because they're slightly different cultures.
Well,
And
in
that's
terms,
what's fun about being an editor in JMSE, because that's places where people actually cross over. That's the kind of stuff I run into.
thank you so much. Yes, and regarding recent research, some studies suggest that microbial networks influence larger marine processes such as nutrient cycling or species symbiosis. So, do you think microbiome dynamics could provide new explanations for ecosystem resilience or decline?
Yes, without a doubt. And that's the future. The future is in the collaborative work between microbiomes and microbial ecologists and then community ecologists. Right now, most of the microbiome work is done on microbiome communities. They're not crossing up to something like an oyster reef or a coral reef and how that's organized. They're not ready for that yet. Um, and we've barely scratched the surface with molecular biology and its capabilities. Um, and that's going to be the future. Functional uh, molecular biology at all levels, all the omics are going to be very important. Uh, right now, if you do one omic, you might not do another. Like you can spend your entire life just doing proteomics. But now I know I need metabolomics. And I'm not afraid to go there because um, there's no silo keeping me out of there. My curiosity allows me to cross into that region. Means I have to work pretty hard because I got to learn new stuff. Uh, and then I get to use it from my perspective. So I'm not a developer of those things, I'm a user. Um, so I use proteomics upside down. I use proteomics backwards. Most people are looking for peptides that they make, not for peptides that someone else makes. Uh, I use enzymes from nature for my proteomics, not pure ones, the real ones. Uh, and most proteomics is done the other way. And it's one thing to be able to say these enzymes are there. It's another thing to say these enzymes are actually active. So lots of molecular biology says, oh, we've got X, Y, and Z enzymes. Which ones are functioning? which organisms are producing them? How do they know? And what are the consequences of those? Those are the kinds of questions I'm interested in. Um, so to me, it's the future of molecular biology. And metabarcoding is just the very first step. And microbiomes are just the very first step in understanding those processes. And we're years away from the capabilities of those systems. And hopefully we'll still have an environment that we can work in when the technology is advanced enough to enable us to do it, and when we can think about how to make it work.
Well, yes, I mean that's it's very interesting you should say that. And there are differing opinions in that regard. And I think one debated topic is whether microbial signatures can serve as bioindicators for environmental changes, for example, pollution and climate shifts. So, what are your views on their detectability and what future techniques or experiments hold promise?
Okay, so we're making a mess of the world right now. Let's start there. Every coastal ocean in the world is polluted. Uh, and we have those signatures. There are people spending their lives looking at algal blooms, harmful algal blooms, uh, changes in the environment that are directly related to nutrients at one level, and then to tons and tons and tons of anthropogenic xenobiotics. Uh, I do a little work on tire dust. So every tire from every vehicle sheds about a milligram of dust every kilometer that the vehicle's driven. And a lot of that dust goes up into the air and it runs off into the estuary. And if you look in that field, you'll find out that um, one of the molecules in tire dust, when it rains in California, leaches out of tire dust and kills coho salmon. And so we're doing this giant unplanned experiment, which is taking our entire communities and exposing them to molecules that change how they all behave. If you look at just different plastic pre-production pellets, the microbiome on every pellet from the same environment depends on the plastic that the organisms are living on. And that's just not instant, that's for years. And so understanding what those consequences are and being able to predict what's damaging an environment, that's going to be one of the first things that happens because people care about water quality. It's one of the few things that people can agree on. I try and do restoration with seagrass. We can't put it back until we know the root causes for why it went away. So when I look at things like restoration, there are short-term things like beach renourishment in the US. You put the beach back because sea level's rising. And there are places in the US where you put it back every year now. That's what's going to happen with other kinds of restoration until we get to the root causes for why it's gone. And a whole lot of that root cause is water quality. And most of it's nutrients. And people keep forgetting that about half the nutrients that come out of the Mississippi River, for example, are from agriculture. The other half are from human beings. And until we actually get our act together and address both of those issues, we won't be putting our environments back. And we're going to end up with mainly bacteria along the coast and the few species that can handle that. So, and we're seeing that. We have a giant dead zone off the Mississippi River. Now, climate change is doing really cool stuff to that too, because if it doesn't rain, the water doesn't flow and you have a smaller dead zone. Or if it rains a lot, the water flows a lot and you get giant dead zones. So for restoration things, you might get seagrass that work really, really well in droughts, 3 or 4 years. And then the jet stream wobbles and it rains hard, ruin the water quality and you kill the seagrass and then you have to start over. So, and having to adapt to climate change is something we're way behind on. And part of it's we don't have the collaborative efforts to make it happen. And the basic knowledge isn't being transferred yet.
Yeah, I mean, you paint um, a very specific picture there, but I'm sure with your research and others that we will definitely find some solutions. Well, thank you so much for sharing your insights with us today, Professor Rittschof. It was an absolute pleasure speaking with you.
Okay, I hope you enjoyed it. I know I did.